SCRF documentary, Dr. Asta interview, roll 04, take 1.
Oh.
Hi, how are you?
Where'd you go to school?
I studied in Utrecht in the Netherlands, where I'm from. I did an undergraduate degree there in biomedical sciences and a master's degree in immunology and virology. And then I moved to Australia, moved to Sydney, and I did a PhD here at the University of Sydney in protein biochemistry.
Okay, now that's nice. Well.
So I actually come from a family of clinicians, and so it would have made sense for me to follow in that path and study medicine, but I didn't. I had a close friend when I was a young teenager, and he was diagnosed with a rare disease. And even though he was given the very best care possible, at the time, from an outside perspective, it still seemed like a bit of a guessing game. And it seemed quite impossible to me that the very best clinicians weren't able to cure him. But those clinicians only have the information available to them to go off of. And a lot of the time, that's just not enough information. And so my friend passed away just after finishing high school. We both finished high school, and it made me think that rather than becoming a clinician, who I see as the executors of medical knowledge, I actually wanted to increase that knowledge and make new therapies possible by becoming a scientist instead. So I studied biomedical sciences for that reason.
Tell me about biomedical sciences. What do you mean when you say that? And I know it should be very obvious with bios and that,
so
the idiots around us.
It's not biology, as in it is biology, but it's not biology that just the whole term of biology. It's a degree specifically focused on training you to become a scientist. So you study life sciences, really. So it's really the intersection of biology and medicine. Anything that would need to be researched in a laboratory is what we study in biomedical sciences. So I'm a person who likes to understand something all the way through and not just a little bit. And clinicians obviously have a very big role in our healthcare system, and they are the people that are at your bedside, and that's hugely important. But they are not the people that are in the lab making new discoveries that ultimately will drive healthcare forward. And that's where I want it to be. I want it to be that person who is really working on creating new knowledge that we can use to make new therapies possible. And that is what a scientist does. There are so many aspects of how our bodies work and how diseases work that we just don't really understand yet. And our role as scientists is to try and understand the disease in as fine a detail as possible to then use that knowledge to create new therapies and new diagnostics. For a while after starting my degree, I wanted to go into HIV research. It was something that really fascinated me, viruses in general, but HIV in particular because of the wreckage it has caused and the devastation, and also sort of the impasse where we were at the time in terms of therapy, like having very good management of symptoms, but still were unable to cure it for the most part. And so that's why I studied virology in my master's degree, and I did work in the HIV field in the Netherlands for a while. But ultimately in science, you kind of go where the science takes you, and sometimes that's different from where you started off at. So while I've stayed with blood diseases, I don't work on viruses anymore, but now I work on blood clotting proteins instead. I work on blood clotting proteins. So those are the set of proteins that are supposed to clot when they need to, and very importantly, not clot when they don't need to. And when there's anything wrong with any of the many proteins that are involved in this cascade, you can get either hemophilia, which is a bleeding disorder, or thrombosis, which is a term for clotting disorders. You don't want either of those things. And these proteins interact with each other in a very refined way. And there's a lot of parts of those interactions that are not fully understood yet. And so we research them to try and increase our understanding of how these proteins work and how they interact. And with that understanding, we hope to improve currently available therapies or create new ones entirely.
Excellent.
I met an Australian, so that's how I ended up in Australia. And I love it here. I love working here. I love living here. I love the lifestyle. I love Australians, really everything about it. And I think Sydney is very special in that it's such a world-class city where we do world-class research, but yet you still get to live in also an amazing place and be near the ocean and sort of be near nature. I think that's pretty special. I love doing this. I feel very grateful to be able to work on new ideas that really have the possibility to change people's lives in the future. And I think that is what science does. So yeah, I would have done research in the Netherlands, probably in a different field, but this is where we are. Yes, so I would certainly say there's a lot of mentorship. There needs to be, because it's a difficult field. It's a highly competitive field. We are mostly dependent on government grants, and so that does make it very competitive. It's also difficult in the sense that there are a lot of disappointments, there are a lot of dead ends. You need to be persistent. My mentor throughout my career has been Professor Phil Hogg, who I saw present his work at the time, and I thought it was so impressive because he's a chemist, and he's trained as a chemist, but he is able to take his discoveries and really bring them through an entire therapeutic pathway and actually get to the point where they affect patients' lives. And I think that's really incredible. So I asked him if I could work with him. So on that, about 10 years ago, we discovered a molecule that we have then put through several iterations and improvements and is now in development as a new cancer therapeutic. So that's something that we're very proud of in the lab. The thing about research is that you often set out to answer a specific question, and of course you try to do that with as much knowledge as you can gather. But still, in answering that question, you're invariably going to get to somewhere that maybe you hadn't quite imagined, or maybe you're going to get to an outcome that's not at all the outcome that you were after. And so that's sort of what happened with this molecule, is that we were trying to do something else, and in doing so, we discovered a molecule that does something useful that we then years later thought, what, maybe that would be actually very useful in a setting of cancer. And so obviously it takes a long time to be able to prove something like that. But once you do, and you get interest from drug companies, and they can help fund the development of a discovery of ours and make it into a therapeutic. So we stumbled upon this by accident, really. And then after this serendipitous finding, we were able to improve on that molecule and make it into something that can be used as a new cancer therapeutic. The point, I think, of everyone who works in healthcare is to improve patient outcomes. And our contribution to that as scientists is the discovery part. So we work in the part of trying to make new knowledge, trying to understand things that weren't understood before. And of course, that isn't easy, and it does involve a lot of dead ends and a lot of disappointments. But sometimes we find something, and I think that's the difficult part, is to realize when you have found something that can actually be useful, that can actually be developed into something that could really help people a few years down the track. And being able to differentiate those findings from all of the other ones, I think that's the difficult part. But it does happen, and it is what makes our job so exciting, and why I'm also so grateful to be able to do this. We start off with a hypothesis. And in discovery science, the hypothesis might be, look, we don't know how this process works, but it might work this way. And then we put a lot of time and effort into proving whether or not the process works that way. But the reality is that a lot of the time, the answer to that question is no, it doesn't. And so now you're a few years later, maybe a lot of effort, blood, sweat, and tears later, and the answer to your question is no, it doesn't work that way. Still don't know how it does work, but not that way. And yet that's sometimes hard to deal with. But you just have to get up, the next morning and think, okay, cross that off the list. We'll move on to the next thing, and maybe we'll get lucky here. And of course, there's, I'm talking about it like it's all just sort of guessing. And of course, these are educated guesses. We try and be as educated as we can before making these guesses. But in the end, you're still working in a space where the information isn't there. That's why you're doing it. You're trying to further the field, as we say. There's nothing worse than trying really hard at something and really being optimistic, and then it all just sort of fizzles out. That's really disappointing, but it's okay. It happens. You just have to keep on going. I think, we do manage to stay motivated. You have to. Like I said, it's a competitive field. We're mostly dependent on government grants, and so that means that you really do have to be on top of your game to be able to stay working in this environment. Yes, so we discovered a molecule that we found is able to bind very tightly to cells that are dead and dying. And that might sound odd to you, but dead and dying cells are actually quite often found in the inside of solid tumors, because a tumor grows more rapidly than it can provide the cells with the nutrients that they need. And so what you get in the inside of a solid tumor is a core with a high density of dead and dying cells. And because of that, in combination with our molecule that binds tightly to these dead and dying cells, we thought that this might be an opportunity to direct a therapy towards those dead and dying cells. And so we've created this combination of the one molecule that, after IV injection, homes to dead and dying cells that are frequently found inside tumors, and then a therapeutic that will bind to that first binder, and that will then direct a therapy right in the location where you want it to be. So our part in this is the discovery part. So we make a discovery, sometimes, hopefully, if you're lucky, maybe a few times in your career. And it is our job to recognize that maybe that might be useful and to bring it to a point where a drug company might become interested in it. And then it is their job to develop that into something that actually can go into patients, because they have the dedicated expertise and resources that are necessary for that. So we discovered a molecule that's able to bind to dead and dying cells, which just happen to be found quite frequently in the inside of solid tumors. So a tumor is a rapid growing mass of cells, and because it grows so rapidly, cells that are in the inside of a tumor often do not get the nutrients that they need to survive. And so you get this core of dead and dying cells inside a tumor. Now, we discovered this molecule, and this is not what we set out to do, but we discovered this molecule that happens to bind very strongly to dead and dying cells. And this is a small molecule, and we have ways of linking this molecule to a therapeutic, say a radiochemical. And so after injection of a radio-labeled version of this molecule in IV, this will then direct the radiopharmaceutical towards the core of the tumor, so really exactly where you want it to be. And the idea of that is that it would have less side effects because it targets the therapeutic directly to the tumor. And we're having some good results with this in early clinical trials. So we discovered a molecule roughly 10 years ago that we found is able to bind very strongly to dead and dying cells. So dead and dying cells are found frequently in the inside of a solid tumor. So a tumor is a mass of growing cells, and it grows so rapidly that it's not able to provide cells that are on the inside of that mass with the nutrients they need to survive. And so what you get is a core of dead and dying cells inside a solid tumor. And because we found this molecule that binds very tightly to dead and dying cells, we figured that if we labeled that molecule with a radioactive compound, which is commonly used to treat tumors, the molecule would then direct that radioactivity specifically to the tumor and then have less side effects to healthy tissues. And so that was the idea, and that has been in early clinical trials, and it's showing very promising results. So this is already at the stage where it is out of our hands. So our part in this is the discovery part. And at the moment that we can show by early experiments, these involve animals, perhaps a few people in like first in human trials. But at that point, this is not the discovery part anymore. This is now clinical development, and that gets handed over to drug companies because they are the people that have the dedicated expertise and also the infrastructure and the capital that's necessary to develop a discovery into a drug. But actually, most of the therapies that are available right now have started off as a discovery made by a scientist in the lab at one point or another. I'm playing with blood clotting molecules, so I work on a few different ones. One of them is called factor VIII, and this is the molecule that's missing in people that have hemophilia A, so the bleeding disorder known as hemophilia A. And the therapy for hemophilia A at the moment is to give someone injections of the missing protein, so of factor VIII. And this is an ongoing therapy, so we don't have a cure for hemophilia A, so this currently requires people to get factor VIII injections maybe twice a week for the rest of their life. Creating the protein that is necessary for this therapeutic is very difficult for complicated reasons that I won't get into. But it makes it that the therapy for hemophilia A is actually one of the most expensive ones that you can have in Australia. And so this represents an opportunity, I think, for us as scientists is to try and research the exact biochemical reasons for why this protein, factor VIII, is so difficult to produce, and perhaps find opportunities there for improving it. The ACRF provides us with the latest technologies that we need to make these discoveries. You can't do good science by yourself. Our expertise is often so specialized that you really need a team of people with different backgrounds in order to do something meaningful. And luckily, we are here in a great environment with a lot of different teams that have great expertise. We work together with the Sydney Imaging Facility at the University of Sydney, who have amazing preclinical expertise. And I think collaboration is one of the necessary aspects of good science, and you just can't really get anywhere by yourself. I also think scientists need to be persistent and dedicated. I think you need to be pretty enthusiastic about, sitting down and trying to understand something as well as you possibly can. You can't be easily deterred. Yeah, I think to be a good scientist means to be passionate about discovery, to be persistent, but also to keep an open mind about what the ultimate goal is. So as difficult as that is in terms of discovery, like you don't know where you're going to end up, but to be able to still keep an open mind and differentiate those important findings that might have a real impact on people's lives from the distractions, I think that's the really difficult part. And yeah, then to deal with the disappointments, I think you need a bit of a distraction. Maybe you need a hobby just to get your mind off things every once in a while. My hobby is surfing. It helps it helps a lot. Makes my mind feel calm. So yeah, I'd recommend it. Trying to constantly be on the cutting edge of science means you have to keep up with improving technologies, and it's really amazing, I think, how far we've come. AI has made a huge impact on the way I work from day to day. Google's AlphaFold, which is an AI-based software that allows us to make computer-based predictions on how proteins fold. And someone like me who works with proteins in the lab and tries to, re-engineer proteins and try and understand how they work, for me to now be able to go to my computer and within a few seconds predict what a protein is going to look like and what maybe a mutant of that protein is going to look like is just incredible. It's really been such an amazing year with those discoveries. And now they've gotten the Nobel Prize as well, which I think is deserved.
Please.
I mostly work on blood clotting proteins. Proteins really are the things that do most of the things in your body. If anything needs happening, it's proteins that do it. But proteins can also malfunction. So genetic mutations often affect proteins. So if you have a mutation in your DNA, a protein is going to be made off of that mold from the DNA. And if that is defective, you might get a defective protein. And if you have a defective protein, for instance, you have a defective factor VIII in your blood clotting pathway, that means that you have hemophilia A, which is a bleeding disorder. So individual proteins malfunctioning can lead to disease. Multiple proteins malfunctioning may lead to cancer. Proteins are really the driving forces behind everything in our bodies, and they're very interesting, and I like studying them.